And Eggan to come and talk. Kevin, you only just got off the plane, is that right? Fantastic. So Kevin is from the Harvard Stem Cell Institute. As a young investigator in stem cell biology, he's received quite a few high-profile awards, and I'm going to let you guys read this. I'm not going to take up any more time. Kevin.
Well, I was excited when I got the opportunity to come speak in this session today and to share our work to a more general audience, and in particular the most important audience, and that is the patients with the disease that we've been studying. So you can imagine my anxiety climbing as I was sitting in SFO last night, and they informed us that the plane that I was going to need to take to get here was being diverted from, into Hawaii instead of SFO, but somehow in the last hour at 2 o'clock in the morning, San Francisco time, they managed to find another 747 lying around someplace. And that got us here a little bit before noon, and so now here I am. So if I'm a little bit discombobulated, that's the solution. But I'm so high on adrenaline that jet lag should not be a problem. So if I can have the first slide. I really felt that it was really my goal and really my purpose in this session to say a few words about stem cells in general, because there's been a lot of conversation about stem cell biology in all fields of medicine, and of course with respect to all neurodegenerative diseases, but of course also in ALS, where not only are there several different trials ongoing, but people have talked about a variety of approaches in which stem cells might be mobilized to either treat or come to know more about this terrible disease. And I'm assuming that they're going to be able to get the slides up there in a second, but I'll just begin by saying that I think there's a lot of confusion about stem cell science and stem cell research, and I think that that's our own fault. That is to say that it's the scientists' fault. And I think a lot of the confusion stems, if you pardon that turn of a phrase, from the fact that there are many, many different types of stem cells. And so I think the best place to begin my talk is really with what is a stem cell. A stem cell is any type of cell that can really satisfy two quite simple definitions. The first one is that it can make more of itself. So in stem cell parlance, this is sort of called self-renewal. Okay, so a stem cell can self-renew. And really, different stem cells have different capacities to renew themselves. Some can only renew themselves just a little bit, and they can only do it inside the body, and we can't renew those cells outside of the body. Others we can grow indefinitely in the laboratory, and we can take one cell and we could literally, with enough time and effort, fill this entire room with them. Similarly, stem cells vary with respect to their second very important characteristic, and that is their ability to differentiate into a variety of different cell types. So a stem cell is any cell in your body which can make more of itself and give rise to other cell types. And so as you could imagine, there are many different stem cells in the body, in different parts of your body, and these cells are critical for normal ongoing homeostasis as the embryo is made, as we are born, and as we age. So if I could have the next slide, because I'm not sure whether or not this is going to work here, I'll try it. Oh, there we go. Okay, great, I can drive it. There are really three primary types of stem cells that you hear about, and I'll say a word about each of them. Probably the most confusing sounding, but also the most powerful in my opinion, and so it's worth paying attention to the difficult terminology, are so-called pluripotent stem cells. They're called pluripotent because they're multipotent. That is to say, they have the ability to differentiate into many, many different types of cells. And the reason this is because they represent a sort of short-circuited version of an early embryonic cell type whose normal job in the embryo would be to make all of the parts of the body. So these are cells that we can grow in the laboratory, and they can, in principle, give rise to all the different cell types in the body. Now the trick is unlocking their potential to be able to do that, and I'll say a word about that. Another type of stem cell that you'll often hear about are so-called cord blood stem cells, which are taken from the neonatal cord after birth. And if I could summarize this whole field of research and give you my frank opinion about these cells, it is that they are sort of supercharged versions of the same type of stem cell that comes out of the bone marrow. So for those of you who've had a loved one or a family member who's had a bone marrow transplant for leukemia, really these cord blood stem cells are very, very similar to those. So in other words, I don't think we should hold out a lot of hope for these cells making motor neurons. Okay, and I, for myself, would certainly have a heated debate with those that would suggest that they could. Okay. Now, there are also so-called adult stem cells. In general, there are many different types of adult stem cells. I think if you were to try to generalize this field, I think a fair statement would be to say that different tissues have different resident adult stem cells. And so just like there is this bone marrow stem cell, which gives rise to all the different blood cells in your body and is constantly renewing your blood every day, there are also stem cells that reside in your intestine and are rebuilding your intestine every couple of weeks. There are stem cells which are in your lungs, which are help rebuilding those all the time. And although one of the most important, I think, discoveries in the last sort of 30 years in neuroscience and neurology is the discovery that there are probably stem cells that reside in the brain, they don't seem to make motor neurons in adults. Okay? And so certainly are neural stem cells. The extent of their activity in the human brain is still, I would say, debatable. But I think the general opinion at the moment is that if they can make motor neurons, they don't do it particularly well. Okay? So for these reasons, in my own laboratory, since we've been interested in leveraging stem cell science to do something about ALS, we've been focusing pragmatically on pluripotent stem cells because we can make a lot of them and because they have the capacity to make motor neurons, which is the cell type which is affected by the disease, as we just learned. Okay, so what are these pluripotent stem cells in a little bit more detail? I would say these are kind of the gold standard for stem cell research in that they're very, very easy to work with in the lab. It's pretty simple to get a graduate student or a postdoc or a technician growing them, and you can make very large numbers of these cells while they retain the ability to give rise to any type of cell in the body. And that's very, very different from most adult cell types, which we really can't take out of the body and grow for any appreciable length of time. Okay? So for instance, that's one reason why they don't take bone marrow out of a person and grow large numbers of blood-forming stem cells to give rise to many people. You need a new transplant every time because we don't yet know how to grow those cells in the lab. Okay? Okay, so where did these powerful pluripotent stem cells come from? And the answer is that there are at least two places that scientists can isolate them from. The first and the most controversial are from very early so-called pre-implantation embryos. These are the types of embryos that are thrown away in many IVF clinics when couples are through with their assisted reproduction efforts. So that is to say that when couples undergo IVF, they'll often make more of these in vitro cultured embryos than they will need, and they throw away the remainders. And many will opt to donate those for scientific research rather than just to simply throw them away. We can then derive these embryonic stem cells from them, and as you'll see, we can use those to make motor neurons and study ALS. Now, another important source, a more recent and I think really exciting source, which dovetails with the conversation that we just had about genetic models for ALS, are so-called induced pluripotent stem cells. This is a very new and exciting discovery that was just made really in the last 5 to 6 years. It's based on an important discovery that happened here on the Pacific Rim in Shinya Yamanaka's lab in Kyoto, Japan, in which after many, many years of studying embryonic stem cells, he had this aha moment where he realized that if he forced adult cells, like easily available skin cells, to turn on genes that are normally only on in early embryonic cells, that it might transform them into embryonic stem cells or a cell type very like that. And because they don't come from the embryo, but they come from an adult, but they have very many of the same properties, we call them induced pluripotent stem cells. And you'll hear scientists banter around things over the course of the weeks like IP cells. It sounds like people are going out for a snack, but what they're talking about are induced pluripotent stem cells. Okay? And these cells, as I said, also can grow in the lab indefinitely and give rise to many cell types, including motor neurons. Okay? Now, this is very useful for research, and I think we should be very excited about this, but the genes that are used to exact this transformation also individually happen to be like a who's who's list of your worst nightmare of cancer genes. And so, although we should be excited about this for research and for understanding ALS, I still think we're a ways off from using these cells to make exact replicas of our own cells to put back into the body. Okay, so I think these next couple of slides, at least for me, will encapsulate what all of the excitement is about in stem cell research in ALS right now. So again, the real, I think, exciting breakthrough was the realization that you could take these early embryos, which again would fit on the pin of a needle and would otherwise be thrown away, and make these cells which can be grown in the laboratory and can give rise to all the different cell types in the body. And I would say that if you're sort of an avid reader of the science pages in the newspaper, you probably realize there's a lot of excitement about, for instance, transplanting these cells using for regenerative medicine. But what you might not have heard as much about is using these to actually better understand the disease. And I guess if there's one thing that I would want you to take away from my talk today is that people in neurology research, and also in this field in particular, are very excited because we think that these stem cell lines, which are going to be either made from early embryos or actually made from patients, are going to serve as sort of flight recorders for the disease. They're going to allow us to replay in the laboratory, in the lab dish, many different events that we think are related to motor neuron degeneration and come to understand it at a cellular level that we couldn't appreciate before. This might not sound like a particularly surprising thing, but if you step back for a moment and you sort of concede that we've never been able to isolate, as far as I know, even a single motor neuron from a postnatal person to study in the lab, we don't know a lot specifically about human motor neurons. And it's almost impossible to get enough, say, mouse motor neurons to do a drug screen that would use the actual cells that are getting sick in the laboratory. And these stem cells will allow us to do that. They'll allow us to play the disease process over and over again in the laboratory, and also begin to do drug discovery in the actual cells that are getting sick. So this is sort of, I guess you could say, counterintuitive from what you might normally think stem cells would be useful for, but I think that in the end, it offers a new potential route towards a better understanding of the disease that we didn't have before, and also a new way to discover drugs in the disease that also we didn't have before. And I'll show you a couple specific examples of what I mean. So of course the other important component of this is that we can begin to make stem cell lines from patients which have those same properties. We've been doing this, for instance, in collaboration with Bob, who you'll hear from next. This is allowing us to make motor neurons from many, many different ALS patients and ask, does the same thing go wrong in all of them? Or is each different form of ALS a slightly different story? Are the things that go wrong in familial ALS the same things that happen in sort of garden variety sporadic ALS? Are there similar problems there? Or are the rules different? And we'll have to think of each one of these as separate diseases that we'll need to treat in different ways. So that's another exciting thing which I think we'll be able to begin to study. Oh, now I'm worried that I'm jumping ahead. Okay, so importantly, this is not just, I guess you could say, a fairy tale. I think for a while there was a lot of promise that we would be able to use stem cells to make large numbers of motor neurons. And for a while we were uncertain how realistic a goal that was. But I would say now it's frankly a reality. And we can take stem cells like this and begin to make millions and millions, maybe even billions of motor neurons in the lab every week. And here you can see some of these cells which have been differentiated from stem cells. They express these proteins which are found specifically in motor neurons. Hopefully you can see this, and hopefully this movie will play. That's what I was fiddling around back there doing. It doesn't look like it's running. But also importantly, as we grow these cells in the laboratory, they become electrically active just like motor neurons. Uh-oh. Oh well, I'm not going to belabor the point if the audiovisual isn't going to cooperate. But those motor neurons, what you would have seen is flashing spontaneous activity, which was indicative of their electrical activity, which is of course a hallmark feature of a nerve cell. Another important thing that we just heard that's unusual about these neurons which are affected in ALS, which is really unique for any type of nerve cell, is that it makes a specialized connection with another type of cell which is not itself a nerve cell, and that is the muscle. And here you can actually see some of these stem cell-derived neurons growing on muscle cells. And although it's a bit bright in the room, what I hope you can see is that places where the arrows are pointing you'll see this red staining. And that's actually indicative of the accumulation of a protein that only aggregates like that where the muscle is connecting to the motor neuron. And so, in fact, these stem cell-derived motor neurons can even make the muscle contract as they do in the body. So these things don't just, I would say, have a passing resemblance to motor neurons, and that's very, very important if our goal is to use them to study ALS where this very specific type of nerve cell is affected. Because if we've made some other type of nerve cell, we might be going down a blind alleyway. So if you go on to, let's see, to the next slide, this is just to show you we can make very, very large numbers of these cells. It's not just that we can make a couple in a dish, but we can really make, in this just one field, thousands and thousands of cells. Okay, so how can we use these cells to study ALS? And I want to show you just two very brief examples of this. This is a nice slide from Jean-Pierre Julien, who's a scientist in Quebec City, and I won't walk you through the details of it, but hopefully just in broadest strokes you can see from these dark arrows that there's a feeling that there are things going wrong inside the motor neurons in ALS, but there's also an accumulating school of thought, as you can see from these sort of ominous-looking lightning bolts, that other cells in the nervous system also in the context of disease start attacking the motor neurons as well. And so one of the things that we wondered was, could we use these stem cell-derived motor neurons to begin to study this toxic interaction between these other nervous system cells called glia, which just stands for glue because they're the other cells in the nervous system, and the motor neurons. And so what we did was to take either these cells from a normal mouse or from an ALS mouse model like the one that was just talked about, and we cultured them with human motor neurons from normal embryonic stem cells. And hopefully this movie will play because I think it's a very nice movie which will show you these motor neurons that are derived from stem cells. This movie is about 2 weeks long. Each frame is 30 minutes in length. And what you're seeing is that over the course of time, these cells which are labeled green are going to make very complicated connections with each other. But that, at the exception of the very, very beginning of the movie, really none of these cells are going to die. This is sort of, I would say, the most supportive environment that we can offer neuronal cells like these in the laboratory. So they're sort of very coddled under these conditions. So unfortunately, like I said, I think that the movies are suffering a little bit, but hopefully you can see that they've elaborated those processes. I'm getting a lot of glare out of the windows. But if you go on to the next slide, what we'll see here, if it will run, is that now we're looking basically right next door on the same microscope stage. It's exactly the same motor neurons, in fact, but now they're growing on a similar set of cells that came from the ALS mouse. Okay? And what you'll see, I hope, if it doesn't start stepping in a similar way, is that although everything seems like it's going to be okay for a while, about a week into the movie, these cells will start to die. And I think that this is, in a sense, an important realization because, again, there's nothing wrong with the motor neurons per se in this movie. It's these other cell types from the nervous system that are changed. Okay? And they're changed somehow by the course of the disease, and we need to understand how that happens. Okay? Now, one reason that we're very excited about this system is that we think it's a new aspect of disease for which there are no current therapeutics. So for instance, as you can see in the difference between the blue and the red bars here, and DMSO is just sort of a negative control where there's no drug. It's just the thing that the drug is normally dissolved in. What you can see is that when we add Riluzole, which is the drug on the market, it doesn't offer any sort of protection from this toxic effect. Okay? But there are many, many other candidate compounds, and these are just sort of generic examples where we can add different drugs, some even approved drugs, where it seems to be able to protect you from this toxic effect of the glial cells. Okay? And so now this is, of course, just cells in a dish, and so I would urge a lot of caution. But it is the actual cell type which is affected by the disease. So the next step is to take these things and to see whether or not they can increase the lifespan in the animal. But it is a new aspect of ALS biology that we haven't been able to really get at before, and I think there's a lot of excitement for that reason. So the other thing that, and these are just my last couple of slides, and then I'll be done, that we're really excited about is to be able to use this as a way to generally get at the question of genetics in ALS in a functional way. Bob and his lab and others like his have been real leaders in this field, discovering new genes which contribute to ALS. I think this has done more than anything else in the last decade to increase our understanding of this disease. But we need some sort of computer for reading out the meaning of these different mutations. And although the mice are useful, it takes a long time to make them, and there are certain limitations to those models that were discussed. And so we need an alternative model which can allow us to get at similar questions. And so what we can do is literally take skin cells from the patients, turn them into stem cells, and then make motor neurons from those stem cells and ask, are they fundamentally different in ways that seem relevant to disease? And this is, I would say, very, very early days for these types of experiments. It was only really 3 or 4 years ago that we made the first stem cell lines from patients. And if you take into the consideration that it really takes a couple of months to make a stem cell line, and then it takes a couple of months to actually make the motor neurons from them, although this is much faster than what we had before, it's still moving along at what sometimes seems like an unsatisfactory pace. But nevertheless, we've been able to begin to make some of the first comparisons between the motor neurons of a normal person and the motor neurons of an ALS patient. And some of the things that we're seeing are really encouraging. And so again, I guess you could say that these next two experiments, contrary to the ones that I showed you, are really focusing on things that we think that are going on inside the motor neurons themselves, which might be contributing to the disease. Okay? Now, because there is the most known about mutations in the SOD1 gene and how they are thought to contribute to ALS, we've begun there. And one of the things which we've seen, which is quite striking, is that when we make motor neurons from patients which have these SOD mutations, they seem to persist more poorly in culture. This is a very I would say, broad finding that they don't survive as well, and there are many things that can contribute to that, but it does suggest that there's something that might be fundamentally wrong from them, even from the very, very beginning. Another thing that we're very excited about is, you alluded to this idea that if anything, it seems like ALS patients might even be more healthy or in some cases more athletic. And one thing which is definitely observed in ALS patients is that their nerve cells are what is called more excitable in the body. That is to say that when you stimulate those cells with electricity, it's easier to get them to fire what's called an action potential. That is to say to actually fire an electrical stimulus that goes and makes the neuron make the muscle contract. And what's great is that that's something that we can actually look at in the laboratory very easily, and so we can ask, is that true about the motor neurons of ALS patients right from the very beginning when they are first made, or is it something which is acquired over time? And at least in early experiments, it really does seem like it might be a fundamental difference in the motor neurons of at least this type of ALS patient. That is to say that patients which have these mutations in SOD1, where now for many different stem cell lines from several different patients, we've seen this fundamental difference in the behavior of motor neurons. And what you're seeing is really motor neurons from 4 different people. The top 2 are 2 normal people, and the bottom 2 are 2 individuals with motor neuron disease caused by the same SOD1 mutation. And you can see that when we apply a similar amount of electricity to those cells, the ALS motor neurons fire more so-called action potentials. And so this could be something which is different from them right from the beginning, and it could offer us an opportunity to come to understand why that is and how it contributes to disease. So again, I think if there's one thing that I'd hope you would walk away from here remembering is that although there is a lot of potential utility very far down the line for taking stem cells and making cells to replace those that are destroyed in disease, there's something very useful that we can do with them right now, and that is to really use them to come to understand more about the disease and to do drug discovery. And so hopefully you can tell your friends that that maybe this will allow us to understand disease like the way the FAA uses the black box to understand plane crashes. And I think I went a couple minutes over, but thanks very much.